Thank you, Kevin, for that introduction and the invitation. And thanks everybody for being in the audience today, and Melissa for organizing this whole thing. I was super excited when Kevin reached out to me to give a talk to this audience. I've attended several of these in the past, and they've always been extremely exciting and illuminating. As Kevin said, today I'm going to be telling you about some of my work that I've done during my postdoctoral fellowship in the lab of Dr. Michael Bruker at the University of Washington, looking at the effects of neuromodulation on amygdala neuron activity. And here I'm in the talk, I'm kind of using neuromodulation somewhat as a double entendre. For the first quarter of the talk, I'm actually going to be going over some work looking at how the neuromodulator norepinephrine is released from the locus coeruleus, or LC, in the basolateral amygdala to promote anxiety-like behavior. And then from there, I'm going to move on to the work that Kevin just introduced, looking at how the basolateral amygdala encodes valence through the use of some new multiphoton neuromodulatory tools to manipulate discrete deep brain ensembles. And when I was putting this talk together, I noticed that probably 90% of the data you guys are going to see today was generating using various Bruker microscopes, some microscopes produced by Inscopics, and then others more recently, two-photon microscopes produced by Bruker. So I feel like it's necessary to say that the only thing I've received from Bruker is lots of help and support so far, so there's no disclosures. Okay, so broadly, I'm interested in how neurons form ensembles, and much of my thinking has been influenced by the work of Eve Marder and others, which suggests that neuromodulation is well suited to dynamically control ensemble activity. And as Eve says in her 2012 review here, that modulators could effectively change the gain of one portion of a circuit or process without altering others. And during my postdoc, I've been trying to experimentally demonstrate what neuromodulatory control of ensemble gain looks like, how it's generated, and its significance to behavior. And as I said previously, I've been trying to answer this question in the context of this highly conserved stress-sensitive circuit from the locus coeruleus, or LC, to the amygdala, the basolateral amygdala portion of the amygdala. And so we know that the LC is stress-sensitive, and that an increase in LC neuron activity to a tonic level of free firing is correlated with poor task performance. Here's just a modified image from Gary Essen-Jones and Jonathan Cohen, showing this kind of modified Yerkes-Dodson curve where we no longer have arousal on the x-axis. Here is now LC tonic activity, and some level of tonic activity is important for optimal task performance, such that phasic activity remains, but stress is thought to promote an elevated level of tonic activity such that phasic activity is kind of drowned out. And so one thing that we wanted to do was understand what activity patterns are important for regulating gain. Then the second question I wanted to examine is, how is this gain control reflected downstream at the ensemble level in the amygdala? What are the cell types and receptors that mediate this ensemble responsivity? And the lab has previously demonstrated that driving this projection from the locus coeruleus to the amygdala is anxiogenic and aversive, though we know very little about the impact of this stress-like activation on BLA neuron activity. Here's just a recording done in an anesthetized mouse by Jordan McCall in 2017, showing kind of diverse responses when driving this neuromodulatory input in an anesthetized animal. So we'll start by answering these questions, and we'll move on to the multiphoton stuff that Kevin talked about at the beginning. And so to answer the first question, we needed a way to record LC neuron activity, and this is what I started in my postdoc, and it turned out to be much more challenging than I anticipated. The LC itself is very small nucleus in the brainstem. It's located at a kind of inopportune spot just next to the fourth ventricle, barely into the brainstem. And in the mouse, it's only about 1,500 neurons per hemisphere, so it's very, very small and located in a difficult position to work with. So when I started, I used the kind of traditional deep brain imaging approach with a standard Grin lens, putting this above neurons infected with GCAMP in the LC. One of my first mice, I received this really beautiful video of these huge adrenergic neurons in the LC. This is Z-stack just zooming through the LC. And this was super exciting, and that we can, optically access these neurons. But you might notice from this video that there's some problems here, chiefly that it's very wiggly. There's a lot of motion. And as it turns out, this type of imaging was really not sufficient to obtain stable recordings within a day, and particularly between days. The field of view would shift. There's a lot of torsion on the neck when mice move around. So this strategy was not going to work very well. So I opted to try a different strategy in which we're using now a prism grin lens directly next to the LC, implanted a bit deeper. But the thing that I thought would help was that the Z motion when the LC was imaged with the traditional grin lens would now be translated into XY motion, which normal motion correction algorithms are pretty good at correcting for. So I hope that this would allow us to better motion correct the signal that we're seeing. And so what we did, and I'm going to go back for a second, this is now a video of LC neurons, about 20 neurons in response to anesthesia. So you see the signal dims here. We taper off the anesthesia, and the signal comes right back. Again, these are neurons thought to be important for arousal, so anesthesia predominantly shuts them off. And so this suggests that the approach is viable. I can tell you from experience, it's much more stable than any other approach. It allows us to record more neurons than I'm aware of have been recorded from before. And this is just a histological image showing this very large 1 millimeter prism grin lens just directly opposed to these DBH neurons expressing GCAMP, these LC adrenergic neurons. And so then we can put mice under the two-photon microscope and start to ask questions such as, what's the effect of stress on LC neuron activity? And so to do this, we put mice under the scope, and we just presented them with a very potent synthetic predator odor 2MT. And then we recorded their activity before and after 2MT. And so here's a different mouse, maybe about 30, 35 neurons in this mouse, and we can see the just ongoing activity. Each of these circles, again, is a putative adrenergic neuron expressing GCAMP. And so we can isolate the activity of each of these neurons and track them as a function of time in response to this predator odor. What we find is that neurons on the whole all kind of display a very similar pattern of activity, that they increase this kind of slowly ramping activity pattern when presented with the predator odor. When we look across the entire population of neurons, this is what we see. So this is data from one mouse. We've now done this in, I think, about 10 mice. And the effect of predator odor is to just slowly produce this ramping increase in LC neuron activity that's highly synchronous across units. And so this ramping effect was quite intrigued by. The hypothesis was, again, that the stressor would increase tonic levels of LC activity. And the ramp, to me, looked like something that would be reflected in kind of a tonic change, a slower change in continuous spiking. And so we used a deconvolution algorithm generated by Daniela and Alana Witten, which was based off of dopamine neuron activity, but we applied this to our LC neuron activity. And what we find is that indeed, during the period in which the predator odor is on board, we see this pretty dramatic increase in deconvolved spiking activity under this GCAMP fluorescence. And this is true across our neurons, that almost all the neurons display this significant increase in tonic activation. And so we wanted to next move downstream and evaluate the effect of this synchronous tonic output on norepinephrine release in the amygdala. And so we did this by